Well, by the end of this session, I hope we'll learn about the process of production and maturation of lymphocytes, the role of different type of T lymphocytes. So there are different lymphocytes in our body, Th1, Th2, Th17. So we'll see what are they, how they are produced first, and what are their different roles, and explain how antigen structure can differentiate between these different subsets, and describe the importance of different factors that trigger the production of these Th subsets. Now, before going into the details, I'll just briefly, recall all the things that we have so far discussed, because a lot of those things will be repeated today. So we started off our first lecture with innate and adaptive immune system. We talked about how innate immune system is a non-specific system. It is very fast. It immediately tried to attack the pathogens. The adaptive immune system was more slower. It is more specific, and it takes time, but once it is done over the period of time, the exposure, the response is very strong, and it can deal with the pathogen strongly. Then there were certain terms like the antigens, epitopes, the antibodies, the 5 classes of antibodies. I talked about IgG, IgA, M, D, and E, and the complement proteins was the previous lecture. We talked about how the complement proteins are activated by the 3 different pathways, whether classical, mannose binding lectin, or alternative pathway. What are their biological functions? Now we'll focus, and one important thing is that in first lecture I told you about the barriers, the physical, biological, mechanical. These were the first line of defense. So once this is breached, then the cellular component of the innate immune system, macrophages, dendritic cell, neutrophil, that is the second line of defense. Then the third line of defense is the adaptive immune cells, which we are going to talk about today. So if we look at the total immune cells that we have, for example, if you measure the immune cells, white blood cells in us, they are about 4,000 to 11,000 per microliter. Now these cells, what are they composed of? From the myeloid lineage, we have got granulocytes, neutrophils, eosinophils, basophils, and from the monocytic lineage, we have macrophages and dendritic cell. On the other hand, the lymphoid cells, they are composed of T cells. T cells have their own subsets. We have already talked about 2, which we'll go in detail today, CD4 and CD8, the helper T cell and the cytotoxic T cell. And there is one more addition to this that is called as the regulatory T cells. While on the other hand, another lymphocyte is a B lymphocyte, and that produces antibodies when it becomes a plasma cell. And finally, the natural killer cell. So if we look at the total number of white blood cells, among those, about 60% are the granulocytes. I'm telling you about the average percentage, and 10% are the monocytes. So 70% is the myeloid lineage. The remaining 30% is these lymphocytes, which are T cells or B cells and natural killer cells. So we'll focus on these cells today. Now here you can see it's just an overall summary of how these cells, in which group they belong and what are their functions. So natural killer cell, it's basically the second line of defense. First line was barrier, second line was macrophages, dendritic cells, neutrophils, and natural killer cell. The third line of defense is B cells and T cells. So it's actually part of the innate immune system. That's why you can see NS is written, non-specific. So natural killer cell are the second line. It is non-specific. What does it do? The name tells you itself, it's a natural killer cell. It will kill viral infected cells and tumor cells. Number 2 is B cells. They are part of the adaptive immune system, and this is the third line of defense. And how it functions, it's called humoral immunity. It produces antibodies, and those antibodies then perform a lot of functions. We'll see about it. Here is something bold for you, except the thymus. So thymus is an organ, all of you must be aware of it. Yes. B cells are not found in thymus, despite the fact that it's a primary lymphoid organ, but B cells are not there. Then T cell, again, it's a third line of defense. It's a specific immune response, and it's actually at the center of the whole immune system. It helps, that's why its name is helper T cell. It kills, that's why one of them is cytotoxic T cell. It regulates the immune response, that's why one of them is regulatory T cell. So they have a lot of functions. They are at the center of the adaptive immune system. Now, certain important terms which you know, these are the facts, and these you will be again needing while you are studying this course. Cytokines. So I'll be repeating a lot of cytokines. What are cytokines? These are chemical mediators, and if I put it simply, they help the cells to talk. So when the immune cells have to talk or they have to perform their function, they produce cytokines. So if it is an order to perform a function, or if it is an order to stop doing something, that mediation, that talking is done through the help of cytokines. Hundreds of them have been identified so far, and they will either stimulate, so the technical term for this talking is either they stimulate or regulate the function of other immune cells. Then the big heading is cytokine, but what are the other names for this? Another name is interleukin. Inter means between, leukin means white blood cells. So they help in talking between the white blood cells. Why? Because white blood cells can talk, so macrophage can talk with a T cell can talk with a dendritic cell, but at the same time, sometimes they have to talk with other cells, you know, like with the epithelial cells or the liver cells. So interleukin will not do this. Interleukin is only between white blood cells. Interferon, so all the cells of the body which have the nucleus, they can be infected by viruses, and whenever any cell, whether immune cell or non-immune cell, will be infected by viruses, they will produce interferons. Interferon alpha, interferon beta, and interferon gamma. Some of them are today used in biological therapies for treating hepatitis as well. So remember, interferon is linked with viral infections, and we'll talk about all the details in immune response to virus. But at the moment, just remember, cytokines help communication between the cells. Interleukin just between the white blood cells. Interferon is released, or they will function when there is a viral infection. Chemokines, so cells have to reach at different places. There is infection in your hand, the T cells or B cells or dendritic cell, they have to reach this tissue. How they will do that? They are called by chemical mediators. And these chemical mediators are called as chemokines that help in the movement of the cell. And this is just a small, you know, diagram that shows that the blue cell, it produces the cytokine, and that blue the cytokines are released into the outside into the tissue, target the cell with the help of a receptor, and the effect is produced, whether they want to stimulate the cell or whether they want to stop or regulate the cell. So all of you are clear about all these terms so far? Now I'll start a story. So far we have been talking all in, you know, components, that what is innate system, what is adaptive, this is the barrier, these, you know, antigen, epitope. Let's start talking about it, all these components in a form of a story. So the first thing is where immune cells are produced. So all are produced in the primary lymphoid organ, and that is the bone marrow. Through stem cells, of course, the progenitor lymphoid cells will be produced. They will give rise to B cell, natural killer cell, and T cell. Now, one question is that why B lymphocytes are named? Can you guess? Excellent. The very good answer is that thymus dependent. I'll explain to you what does that mean. And B is, you should remember it as bone marrow, but the reason for this is not bone marrow. Actually, it's bursa of Fabricius. This is an organ in the birds where B cells were first identified. So because of that, they name it B cell, but it fits very well with humans because these B cells are produced and matured in the bone marrow. Now, another question is, what might happen to a person born without a thymus gland? So thymus is a secondary lymphoid organ or primary? Primary. So if somebody does not have a thymus, what do you expect? Digeorge syndrome. Excellent. What does that mean?
There is no thymus, and thymus affects on the parathyroid action.
Okay, so one is the developmental part of it. So they will have any problems with the jaw, facial, looks and heart, and then something happening with the immune system. So we'll see now, one by one, we'll see what happens. I'll try to explain all these things. But we'll go first here. So here you can see B lymphocytes are produced in the bone marrow. T goes somewhere else, B goes somewhere else, lot of terms. So we'll try to catch a story today. So in the bone marrow, T cells and B cells are produced. Their development take place here. Then there is another word for them. Where does they get mature? So T cell will come out without being mature. Okay. So here the T cell that comes out, it is immature. Okay. While the maturity of B cell, it is developed here and it is matured inside the bone marrow, so they get matured inside the bone marrow as well. They have to go further to thymus. Inside the thymus, these T cells are going to get matured. Now what does that maturity means? They will acquire certain type of receptors here. Okay, I'll just mention two of them. So after this maturity, we have got two different type of cells, CD8 positive and CD4 positive. How this maturity take place? Another lecture in the third week. So far, just you remember, they come out of the bone marrow as immature T cells. They enter the thymus. They become mature. When they come out, now what is the term for them? They are called as naive, mature T cells. What is naive? I told you, yes, please. Different?
No.
Naive, anybody knows what does naive means?
Didn't encounter.
Excellent. Any cell that has never encountered any pathogen before in life. Okay, so when it is produced, it is newborn, never seen any antigen before, pathogen before. So, and if you remember in one of the lectures, I explained to you the big diverse number of cells that we have. So around 9.1 million, I told you, can be the B and T cells. So out of those, just couple of them, for example, if we are talking about malaria or typhoid, so for typhoid there are, let's say, 2 or 3 cells that recognizes it. Okay, so how out of that 9.1 million, one is going to go and recognize the pathogen? So we'll try to build the story. They come out as immature T cell, go into the thymus, come out as naive, mature T cell. Now where they are going to go? They are going to go into the peripheral lymphoid organs. So here is a lymph node. Now these naive, mature cells are going to come here with CD4 or CD8. Do you remember the dendritic cell? They are called as antigen presenting cell. Their job is to capture the antigen and take it to the lymph node and present it to T cell. Now, the first time when a naive mature T cell encounters the antigen, it will be with the help of dendritic cell talking to a T cell. By the way, how this dendritic cell is going to recognize the pathogen? With which receptors? PRR. They recognize the pathogen, now they are going to present it. How it's going to present it? MCH, very good. So if it is CD4, it will be MHC class 2. When this happened for the first time, the word for this is called as priming. Okay, so one new term is naive cells who never encountered any pathogen. They entered now the lymph node, and here they are presented the antigen by dendritic cell through these receptors. This process is called as priming. When they are primed, then what is going to happen? The T cell is no more now naive mature, it will become activated T cell. Okay, so the next stage is after priming, it will become activated. Now the next question is, should it become a Th1, Th2 regulatory T cell? It has to change into one of these cells. This process is called as differentiation. So this T cell, CD4 cell, it is going to change into one of these cells. It means converting into some other type of cell. That is called as differentiation. So now we assume that one antigen, let's say typhoid, was encountered by this T cell. There were only few T cells that can recognize typhoid. Those cells are primed. It means they encountered the antigen. They are activated. How the activation take place? What are the steps? We'll see later. Then it will change into that cell that can kill the typhoid pathogen. So it will differentiate into it, and now these cells have to increase in number because we need lot of them. There are, for example, couple of them, 2, 3 that have recognized it. Now we need thousands of them. So they have to multiply, increase their army. What is that called? Proliferation. Okay, so they have been activated, primed, activated, they differentiated into a special group, they are proliferated, the numbers have increased. Now they have to go to the place where all this happened. So wherever the pathogen entered, let's say this is the tissue, and here is the typhoid pathogen. They have to come here, so this big army of cells. It has to move to the tissue. So far we are all good that they are produced in the bone marrow, that is called as development. Then they come out as immature T cells. The maturation take place in the thymus where the certain receptors will come. After that it leaves the thymus as naive mature T cell. These naive mature T cells then have to go to the peripheral lymphoid organs. Most important is lymph node in case of T cells and spleen in case of B cell. Here they encounter the pathogen through dendritic cell which has brought it here, and this process is called as priming when it is done for the first time. Now after priming, the T cell gets activated. Now it has to become either Th1, Th2, Th17. This is called as differentiation. Once the differentiation has occurred, they have to increase their army. Their numbers have to increase a lot. This process is called as proliferation. These proliferated cells, with the help of which cytokine they have to come here? Chemokine, excellent. The chemokine is going to call these cells towards the pathogen, where the pathogen is. Once they kill the pathogen, then the big army of cells, they have to reduce back. You will kill the remaining cell with the help of apoptosis, which is programmed cell death. Now the pathogen is destroyed, so you kill the extra cells, and just couple are left as memory cells for the future encounter. So if you remember, I told you adaptive immune system is very specific. It will only, these cells only recognize typhoid, okay, Salmonella typhi, and once it finishes it, then the memories stay longer for years. So the next time Salmonella typhi comes, this whole process does not need to take place. This memory cells will quickly multiply and immediately attack. So that is the reason for the future encounters to be very fast. They are very quick. But for the first time, priming, it takes weeks, sometimes 14 days, sometimes 2 weeks for this whole process to take place. So far, any questions? Okay, so now in this slide you can see that bone marrow has produced the B lymphocyte, T lymphocyte. T lymphocytes go into the thymus, B lymphocytes stay in the bone marrow and they get matured. After that, this secondary lymphoid organs come into play. For example, this lymph node, where they will get activated. And what are the other secondary lymphoid organs? Spleen, mucosal and cutaneous lymphoid tissues. And as a result, you will have, this is the cell-mediated immune response, okay? But if the B cell come into play, that is the humoral immune response. Now I'll just quickly put some steps of B cells as well. So B cells come out as immature cells or mature. So of course these B cells that come out, they have got their B cell receptor. This is called as BCR and this is collectively called as TCR. So these B cells, they are going to come as well to the spleen. Mainly spleen has got majority of the B cells. Now, one more thing I want to clear it. We couldn't finish it in our one of the lectures. I told you there are 3 cells that have got MHC class 2. Macrophage, dendritic cell and B cell. You remember that? Now, they all are called as antigen presenting cells. But they all have different functions. They do everything for a different reason. And I'll now explain all 3 different functions now. Dendritic cell has got PRR, it captures the pathogen, present it to T cell for the purpose of priming, first time encounter, okay? The macrophages are not circulating in the blood, they are always in the tissues. So there is one macrophage here, for example. This macrophage, with the help of PRR, will recognize Salmonella typhi, engulf it, and then wait here. Okay, for what it is waiting? It is waiting for the activated T cell to come here. Then this time when he presents the antigen to T cell, what is the purpose? They ask for permission to kill the Salmonella typhi. So the purpose of dendritic cell was to prime the T cell. The purpose of macrophages to kill the Salmonella typhi which is inside it, but it will always take permission from the T cells. It cannot just engulf and start killing, no. And it becomes effective killer after it takes permission from the T cell in the form of cytokine. So for example, interferon gamma, that will tell the macrophage, kill this pathogen. Now, yes please.
You said that between leukocytes and interleukins are
Yes.
But you said now interferon.
Interferon gamma is the name of one of the interleukin. So there are a lot of TGF beta, interleukin 1, 2, 5, 7, interferon. They are all interleukins. They function between the white blood cells, but they can have different names. Now B cell. B cell is also going to present the antigen to the T cell inside the spleen, inside the lymph node. But what is its purpose? So I told you there is a class of 5 classes of antibodies. IgG, IgA, IgM, E and D. IgM is the first antibody that is produced. You know, the first thing that the body starts is produce IgM. But then according to the pathogen, B cell has to switch This is called as, so the new term is class switching. B cells stop producing IgM, it now start producing IgG, so that the bacteria Salmonella typhi can be killed very well. Now this class switching does not take place automatically. This B cell is going to present the antigen to T cell. Again another interleukin will be produced, which will tell the B cell, stop producing IgM now, start producing IgG. Okay, so if now you compare the purpose, dendritic cell helps in priming, macrophages have to kill the pathogen, B cells have to class switch, they have to change from IgM producing antibody to IgG producing antibody. Each step it is presenting the antigen, so they are antigen presenting cell, but the purpose is totally different. So is this clear to everyone? Yes.
Can you repeat the class change?
Okay, so let's say there are these B cells, lot of B cells. All of them will produce plasma cell, and the plasma cell is going to produce, let's say, IgM. Same. But your body now need IgG. Body don't need IgM. So this B cell has to change, convert from IgM producing cell to IgG producing cell. This step is called as class switching, because one class of immunoglobulin is changed into another class. Class switching, okay? How it will happen? With the help of the T cell. So when they meet together, T cell will tell it, now stop it. With the help of some cytokines, the genes will change and it will start producing IgG.
Class switching is the same like isotope switching?
Yes, it is the same thing, yes. But there is a lot of detail to it that will be discussed in humoral. So I will tell you now, this has a separate lecture about it. Tolerance, lymphocytes, friends and foe. The B cell class switching and how they become specific, that is in humoral immunity. There is full lecture about this. So slowly these things will be explained in detail, but you should know the whole story now. I'll just go through this again. So the bone marrow, it is producing T cells and B cells. T cells come out as immature T lymphocytes, okay? The B cells get matured in the bone marrow and they become mature, naive B lymphocytes. Then the T cells go, immature T cell go to the thymus. In the thymus they get matured. Maturation means they will have some special receptors around them, especially the T cell receptors. Then they come out as CD4 cells or CD8 cells. These CD4, CD8 cells will go to the different secondary lymphoid organs, spleen, lymph nodes, blood. Here when they enter the lymph node, the dendritic cell that has captured the pathogen, it will present it for the first time to a T cell, which is called as naive mature T lymphocyte. And the first time presentation is called as priming. Once it's primed, it has to become now active. Activated, we will see how is activation taking place. Then it has to become one of the, yes please. So when it goes into the spleen, it has to encounter a T cell. Same process like this. It will encounter the T cell and the T cell will convert it. It will become active. And then it will start producing either IgG, IgD or IgE, and those antibodies are then going to perform its functions. So that end result, their differentiation or end result is you want lot of antibodies. So that's the function of the B cell is performed by the antibodies, but the function of the T cell is performed by the helper T cell, cytotoxic T cell and natural killer cells or regulatory cells. They perform the function of the T cells. Then a big army is produced. That big army is called, this production is called as proliferation. And finally all these cells with the help of a cytokine will move to the tissue. in the tissue, they will try to kill the pathogen, and also it is going to help the macrophages, which is going to present it antigen in the tissue. And the T cell is also going to help the B cells, which will present the antigen to the T cell. So they will all together now produce antibodies, helper T cells, cytotoxic T cell, and they will try to kill the pathogen. Yes?
How T cell will